Yeah, it's blinding. Okay, why don't we get started? Welcome back to session 9, the building blocks of cognition. As is customary for the last talk session of the day, we're going to start with selected talks before finishing with a special lecture. And our first speaker is Lucas Tian from the Rockefeller University. He's going to talk to us about neural substrates of a symbolic action grammar in primate frontal cortex. Thank you.
Thank you to the organizers for this wonderful conference and for giving me the opportunity to present our work on neural substrates of a symbolic action grammar. Imagine how you would respond if I asked you to draw an animal that doesn't exist. Children around the age of 9 are actually quite good at this task, and interestingly, they tend to use a strategy of recombining learned parts in novel ways. This includes mixing parts from different animals, such as a pigtail and a camel hump, and repeating a part, such as a head, a variable number of times. This capacity to systematically recombine discrete components into composite representations is called compositional generalization, and is thought to underlie many cognitive abilities, including in language, reasoning, action planning, and imagination. But its neural mechanisms remain poorly understood. We hypothesize that compositional generalization depends on internal mechanisms reflecting symbolic grammar. Grammars consist of discrete elements, or symbols, and rules, or syntax, for combining these elements into a large set of potential representations. To illustrate, consider the following, the drawing task. An appropriate grammar might consist of symbols representing categories of body parts, and rules for combining these parts, such as repeating a given part n times. Critically, there exists empirical evidence for internal grammars, including from behavioral patterns and how people and animals generalize to solve novel problems. There remains, however, a major gap in knowledge because whether and how neural activity implements symbolic grammars is unclear. To move forward, what we need is an animal model involving grammar-based generalization. We're excited to report that we think we have succeeded in doing just that in the form of a macaque drawing task. This task paradigm builds on foundational work, especially on cognitive structure in drawing and handwriting, including our 2020 study of human drawing, and research into the neurophysiology of motor primitives and sequencing in non-human primates. At the surface level, this task is simple. Monkeys are shown images of composite geometric figures and have to draw them by tracing. The critical aspect is that they are constantly challenged with novel images. To draw these images, they draw these images using a learned action grammar with two components. Stroke symbols, which are action categories corresponding to single strokes, resembling shapes such as a line or a circle, and sequencing rules, or syntax, which defines ways of combining strokes, such as connect a line to a circle to make a lollipop. We have found that they can generalize to draw new images, and that they do so by reusing this grammar. Here are some example images drawn by one monkey. You can see that the monkey is making drawings that resemble each target image, but interestingly does so in a way that is not directly tracing, but instead reflects the monkey's constructed version of that image. As I'll elaborate later, this interpretation of the monkey reflects a composition of a small set of learned stroke symbols, each of which is represented here with a different color. In this example trial, the subject initiates the trial by holding the blue button and has a couple seconds to observe the image and plan. After a go cue, he can draw using any number of strokes of any type. He decides on his own when to end by pressing a done button, at which point he gets juice reward proportional to the spatial similarity of the final drawing to the image. And so here's another trial from a different monkey, Diego. So establishing this behavioral paradigm gave us the opportunity to investigate the neural basis of action grammars. For this talk, I'll focus on the identification of a neural representation of action symbols in a localized area of frontal cortex. This is work that we recently submitted and have posted as a preprint. So in trained monkeys, we performed simultaneous recordings across multiple regions of frontal cortex. We implanted 16 32-channel arrays, targeting many areas, including areas associated with motor control in M1, with action planning in premotor cortex, and higher cognitive functions in prefrontal areas. PMV, or ventral premotor cortex, or F5, is highlighted here because it is the area where we identified a representation of action symbols. We focus on three essential properties of putative symbolic representations: invariance, categorical structure, and recombination. We asked whether we can identify these three properties in both behavior and neural activity. For each of these properties, I'll describe behavioral evidence, as well as neural evidence for encoding in PMV. So first, invariance. Invariance means that activity exhibits a form of abstraction in which it is largely independent of variables irrelevant to the task. A well-known form of invariance in handwriting is the phenomenon of motor equivalence, in which writing exhibits trajectories invariant to specific muscle patterns being used. Inspired by this, we test the generalization of stroke trajectories across motor variation due to size and location. The left schematic depicts 3 images. One is a well-practiced circle, and the other 2 are novel combinations of size and location. The right depicts possible drawing outcomes. First, the monkey may simply fail to generalize. Second, he may optimize overall movement efficiency by taking the shortest path, even if that means that each stroke trajectory varies across trials, such as in its starting location or whether it's clockwise or anti-clockwise. Third, he may exhibit motor invariance by reusing the same stroke trajectory, even if that means taking longer, less efficient paths. We found that drawings exhibited motor invariance. This is clear in 9 example trials for the circle shape across 3 sizes and locations. Similarly, for 4 other shapes, the stroke trajectories were invariant to size and location. We next ask whether neural activity encoded stroke primitives in a manner exhibiting this invariance over location and size. We focus on the time window between image onset and go cue, which we call the planning window. This depicts an example unit in PMV showing a raster plot and trial average firing rates. Soon after image onset, the student's activity strongly differs by primitive. We can group trials into 4 columns for the 4 different drawing locations. And this reveals that the unit encodes primitives in a similar manner across locations. We summarize the strength of primitive and location encoding across units and sessions. Primitive encoding on the y-axis is a normalized Euclidean distance metric that captures the extent to which population activity systematically varies with primitive, controlling for location. Conversely, location encoding reflects how much activity differs by location, controlling for primitive. Consistent with the example unit, PMV activity strongly encodes primitives and weakly encodes location. Importantly, we recorded across all areas simultaneously and therefore can perform identical analyses for all of them. Doing so, we've seen that PMV is the only area that exhibits strong primitive encoding and variant to location. Next, we looked at categorical structure. To illustrate this idea, consider the simple problem of writing this letter. What about now? Or now? So this example illustrates that because letters are represented as categories, the same image is likely to be interpreted as either an H or an A and not something in between. We performed an experiment similar to that example. We presented images that across trials were randomly sampled from a continuum that varied parametrically between 2 well-learned shapes. Here images 1 and 7. This monkey had already learned stroke primitives in response to these 2 images, which are called primitive 1 and 2. For each trial, we quantified its relative kinematic similarity to primitives 1 and 2 using a primitive alignment index ranging from 0 to 1. We found that subjects drew intermediate morph shapes using strokes that resembled either primitives 1 or 2. I highlight 2 behavioral findings indicating that primitives 1 and 2 were represented as categories. First, there was trial-by-trial variation between these 2 primitives given the same image number 5. Second, there was steep, seemingly discontinuous sigmoidal nonlinearity in the primitive alignment scores, even though the images themselves were morphed in a linear manner. We asked whether neural activity in PMV also exhibited this categorical structure. Here we plot PMV population activity in its space spanned by its first 2 principal components. Each curve represents average activity for one of the morph shapes. After image onset, marked with an X, the activity diverged to 2 states, consistent with categorical encoding of primitives 1 and 2. This was true also for the ambiguous image number 5. And so here we see the activity reflects what primitive category the subject is planning to draw, even for the identical image. And we summarized this effect across brain regions and sessions, found that it was strongest in PMV, and surprisingly did not observe this effect in prefrontal cortex, a region known to be important for decision making and categorization and other tasks. Next, we looked at recombination. We tested whether subjects spontaneously recombined stroke primitives to draw novel images. These images, called characters, were generated by connecting shapes in different ways. The reason that subjects may respond with a few possible kinds of drawings. First, they may fail to produce accurate drawings. Second, they may succeed, but do so by tracing with a single best-fitting trajectory. Third, they may use multiple strokes, but strokes that do not resemble their primitives. Finally, they may recombine their primitives. Here's an example image on top, and on the bottom, the learned primitives for subjects 1 and 2. Subjects 1 and 2 each successfully drew this image, but did so using different strokes. Subject's 1's strokes matched his own primitives, and subject 2's matched his own primitives. Across hundreds of unique images, around 80% of strokes match each subject's own primitives, while only around 10% matched the other subject's primitives on identical images. So this finding indicates that subjects indeed recombine their primitives to draw novel characters. Does PMV activity also reflect this recombination of stroke primitives? For each primitive, we compared when it was drawn in character tasks to when it was drawn in single-shape tasks. Each color trajectory here is the average population activity for a stroke primitive aligned to stroke onset. And it shows that activity for each primitive was similar in PMV between character and single-shape tasks. And summarizing this effect across sessions, we found that PMV population activity scored highly in primitive encoding and low on task encoding. And this effect was strongest in PMV, indicating that PMV activity from primitive is reused in character tasks. Taken together, these results reveal invariance, categorical structure, and recombination in both behavior and in neural activity in ventral premotor cortex. In summary, we established a macaque drawing task to study compositional generalization guided by an internal action grammar. In multi-area recordings, we've identified a localized representation of stroke symbols in PMV. And briefly, I'd like to highlight a few implications. First, this provides evidence at the level of neurophysiology for an internal symbolic representation, a kind of representation thought to be critical for cognition, but whose evidence has largely been inferred from behavioral and computational studies. Second, contrary to our initial expectation that stroke symbols might be encoded in the lateral prefrontal cortex, we found they're represented in ventral premotor cortex. And we propose that prior observations of invariant representations in PMV, including of mirror neurons, may potentially reflect PMV's general role in encoding action symbols. And third, our findings indicate that understanding internal grammatical processes will be critical for explaining motor behavior, especially the flexible generalizable kind. Lastly, although this talk focused on action symbols, which is a core component of action grammars, a deeper understanding of grammatical processes also requires explaining how neural activity implements sequencing rules to systematically compose action symbols. Finding action symbol encoding in PMV provides a foundation for asking how neural dynamics in PMV and interconnected areas may implement grammatical operations. And we're currently addressing this question by combining analyses of behavior, neural activity, and computational models. With that, I would like to thank various funding sources and people involved in this project, including Ueli Liu, who is at this conference and with whom I'm collaborating on a computational model of grammatical rules. I'd like to thank you for your attention, and I'm happy to take any questions, either now or later offline. Thank you.
We have time for a few questions.
Thank you. So my question is basically, okay, so I want to test this specific way, whether it's really learning the symbol. So can you actually test on different hand and like a train on one hand and then block, like just keep the one hand off and then making the other hand? And do you going to see the same?
Yeah, that's a very interesting question. So we haven't done that. And and the main reason is that there is kind of a basic component of learning the basic motor skill to even just put a finger on a touch screen and trace that the monkeys don't naturally have. And so it's not easy to ask them to just simply use your other hand. But that is the kind of experiment that we're interested in doing.
Hey, yeah, great talk. I'm wondering if you could say a little bit about the computational model. Is it like a program induction thing, or are you able to make a neural network do this?
Yeah, we're interested in integrating all these levels of representation. So we do think that something like program induction is the way to go, but we also are really interested in how that might be implemented in neural processes. And so we have some ideas there we can talk about offline, but it's some sort of hybrid thing that we're thinking about.
Do you think a vanilla or RNN or something can do this? In the way that the
I think largely unstructured vanilla RNN in our hands has not really been able to do something like this. We've had to build in various kind of symbolic representations in it, but we can talk more about this if you're interested. Yeah.
Time for one more question.
Hi, great talk. I would like to ask whether you've seen something in the population dynamics of the specific selective region. Like, when you see the composition, do you see overlap in the regions, or just first one, then another? Did you see something, or can you share something related to that?
You're talking about
the specific primitives,
yeah.
the specific symbols. How do you see them imprinted in the population dynamics of the selective region?
You mean if they're like multiple symbols, or what do you mean?
No, yeah, like on a population level, do you see first one, like the corresponding neurons firing for one stroke and then another, Do you see some kind of overlap, some multiplexing? How do you
So in the particular reason I highlighted, particular region I highlighted, PMV, it does seem to have largely a representation of one primitive at a time. But we see some interesting kind of switching and other kinds of representations during kind of the planning phase of the task. Yeah.
Thank you.
Thank you.
Okay, let's thank Lucas. Our next speaker is Tim Sainburg from Harvard Medical School, and he's going to talk to us about discrete actions are a unit of both behavior and evolutionary selection.
So Konrad Lorenz and Nicholas Tinbergen were not neurobiologists, but they put forth one of the foundational theories of neurobiology. The theory was about the modularity of behavior, and I'm showing you a figure of Tinbergen's model of how behavior is generated. At the top, we have the highest level drives, which flow down to the lowest level motor impulses. But what I want to focus on today is this little line in the center of the model. In Tinbergen's model, everything above this line is in competition, while everything below this line is done in coordination. For example, in Tinbergen's stickleback fish, a large portion of its time is dedicated to reproductive behavior. That reproductive instinct drives the selection of complex behaviors, like whether to fight or nest or search for food. And high-level actions drive the selection of low-level actions, like whether to bite or to charge or to scratch during a fight. Tinbergen calls these lowest-level actions consumatory acts, but what they really are atomic behavioral modules. Because beneath those modules, rather than competing, motor systems coordinate to produce a chosen action. Tinbergen and Lorenz never fully agreed on what constitutes an atomic behavioral unit, but they did agree on 3 defining features. And they happen to be the same features we use to define a syllable of birdsong. Syllables are distinct in time, they're stereotyped, and they're syntactically organized. And so what is a syllable of behavior? This is a short clip of a mouse behavior in an open field, and I'm plotting the egocentric positions of the mouse's joints below that mouse, as well as a self-similarity matrix of kinematic features over that time span. And so you can see this block-like structure, and these blocks are what I mean by discrete in time. At around 3 to 4 hertz, mice switch their behavior to a new syllable. If we now take those syllables, we can compute a graph over their kinematic features, where each node represents a syllable, and each edge represents the similarity between syllables. And if we extend that graph to look at millions of syllables simultaneously, we find that clusters emerge. Those clusters correspond to distinct syllabic categories, things like jumping and rearing and locomotion. And finally, those syllables are syntactically organized. On the right, each line here is depicting a transition between 2 syllables, colored by the outgoing syllable type. And on the left, we have an abstraction of that syntax, where each circle is a syllable, and each arrow represents a transitional probability. And so together we observe that mouse behavior is discrete in time, it's stereotyped, and it's syntactically organized, and therefore syllabic. And so Tinbergen's model captures a theory about the modularity of behavior. At the lowest level, we have a set of behavioral modules, which are selected on the basis of higher-level behavioral programs. And those programs are selected on the basis of even higher-level drives. And all of these things are in competition: hunger versus thirst, fight versus flight, scratch versus bite. But that model also has implications for how behaviors evolve. Animals adapt their behavior and their physiology to adapt to niches in their environment. And so 2 species of mice can emerge, for example, such that one specializes to life in the forest and another to life on the prairie. That first species might need to learn to be good at climbing trees, and that other might need to know when to hide from predators. And so an open question in evolutionary neurobiology is how our behavior and its underlying neural circuitry adapts to form those niches. So Tinbergen's model provides a hypothesis space on which we can test how behavior is selected. For example, within a syllable, the kinematics could shift to support new behavioral niches. A mouse predated by owls might produce a more cautious gait. Mice might also develop or lose syllables. For example, a grassland mouse might develop a new rear to sniff out grub. Or selection might occur on higher-level behavioral states. For example, a more territorial mouse might crank up the preference for fight over flight. And so to investigate how syllables evolve, I'm taking a phylogenetic approach. So for this experiment, I've sampled 18 species and subspecies of mice from 2 main taxa, Peromyscus, also called deer mice, and mus. These colonies were caught anytime between the 1970s and 2021. And deer mice were sampled from all over North America. They occupy various ecoregions and ecological niches. I plotted our samples here. And musk can be found all over the world. Our samples are also plotted here. Most of you are familiar with lab mice. Lab mice are usually derived from Mus musculus. Mus musculus live in human-adjacent environments. But other species of mus, like Spretus and Caroli in our data set, live in more natural settings. And so for each mouse, I sample their behavior in a 30-minute open field assay, where I record various aspects of their skeletal and their movement kinematics. And so the first question I want to ask with this data set is whether syllable repertoires are shared across species. So we take our full data set of syllables, and we can break them out by species. So here each color is a subspecies. Unsurprisingly, we find that each species produces each coarse syllable type. They're all rearing, they're all locomoting, they're all grooming. But we can also get more granular. So we can look at not only rears, but sub-rears and sub-sub-rears. And if we now look at these very granular syllables, we're able to ask, are those granular syllables species unique? And so we can quantify this by looking at the percentage of overlap in syllable repertoire between mice. So two mice sampled from the same subspecies have an overlap in repertoire of these very granular syllables of about 67%. So that's our baseline. And as we get further out in phylogeny, we observe only a minor decrease in syllable repertoire overlap. And so this is telling us that syllable repertoire, even at this very granular level, is deeply conserved over those 50 million years of evolution. Although syllable repertoires are largely shared, mice differ in how they employ those syllables. So I've sorted those most granular syllables by those preferred by mus versus those preferred by Peromyscus. And you can see that different syllable types are preferred by mus or Peromyscus. For example, jumps are much preferred by Peromyscus than mus. And so in fact, a linear model trained on syllable distributions can accurately distinguish between two closely related subspecies of mice. So here I'm plotting a confusion matrix where the diagonal reflects a correct prediction, and really the only mistakes that we're making are these very closely related subspecies of Peromyscus maniculatus. And so to provide a concrete example of that syllabic difference between mice, we can look at locomotor syllables. So here I'm showing you two different gaits, which constitute two different syllable types. We have a trot in which the hind legs are in antiphase, and a bound in which the hind legs are in phase. And so when we look at trots, we see that, well, each species trots. Some species trot more than other species. So for example, C57s really like to trot. When you look at Peromyscus maniculatus, that blue subspecies and subspecies here, you see a lot of variation. So some subspecies really like to trot and others really don't. Meanwhile, in bound, there's a preference by just a few subspecies for bounding. And so if we zoom out and we look at all the syllables, we come to this broad observation. Behavioral similarity is decreasing as a function of phylogenetic distance. And so syllable usage is basically steadily diverging as phylogenetic distance is increasing. And so knowing that syllable usage has a strong phylogenetic relationship, that allows us to then estimate how syllables are evolving. And so to this end, I fit a generalized linear mixed model to my behavioral distributions, which capture phylogenetic relationships as a random effect. And so the covariance matrix of that random effect captures which syllables are co-evolving. Some of these are very intuitive. So for example, left and right turn syllables are co-evolving. Mice that spend a lot of time circling around the arena counterclockwise are also gonna circle around the arena clockwise. But we also see less clearly related and more heterogeneous groups. So rear syllables are co-evolving with head movement syllables, or co-evolving with locomotion syllables. And so finally, I want to ask what these groups of syllables are and why they're co-evolving. And so to answer this question, we can take a step back and look at a whole behavioral recording. This is a 30-minute ethogram from a deer mouse. And just by looking at this ethogram, you can see that mice occupy behavioral states that extend over tens of seconds, even minutes. So you can just see that it just left that groom state right there. And so when we apply a learning algorithm to infer those states, we find that there's a few basic states that the mice occupy. I've just post hoc named those states here. And so to help you visualize those states, we can look at them in terms of the UMAP syllable plots from before. So we observe that each of these states involves different syllables, and they involve different syllable syntax. Further, each different species and each different subspecies samples those behavioral states in a different way. So for example, while basically all of the mice are producing this groom state, the mus spretus are sampling from that state much more often than the mus caroli are. And so having inferred these higher order states, we can now ask whether these higher order states co-evolve. And so to that end, we look at the relationship between the coevolution matrix and the higher order states. And so to visualize that, I'm computing PCA over the coevolution matrix, and each dot here corresponds to a syllable. Syllables that are closer together in PCA space are co-evolving. And so we can color those by the higher order behavioral states that each syllable occupies. So for example, these orange grooming syllables are almost always produced during the grooming state, and they tend to co-evolve together. And we see this for each of these different states. And so this suggests to us that one way in which behaviors are being selected is on the basis of these higher level behavioral states. And so to summarize what we found, syllables are discrete, they're stereotyped, they're syntactic, they occur in both wild and lab mice. They're deeply conserved over 50 million years of evolution. Behavioral distributions diverge slowly over evolutionary timescales. And finally, behavioral repertoires appear to be selected for in a modular fashion, both as syllables and as higher order states. And so bringing this back to the Tinbergen model, our observations provide support for behavioral modularity and suggest that behavioral modularity serves an evolutionary purpose. Syllables remain stable over 50 million years of evolution. Within syllables, we see some adaptation. Part of that adaptation might be in repurposing syllables, and part of that might be accounting for things like changes in body morphology and physiology. But the independence of these modules is allowing for rapid adaptation. And so together, these behavioral observations provide a lens through which we can now investigate how genomic changes modulate neural circuits and how those in turn modulate behavior. So with that, thank you for listening. Thanks to my collaborators, Andy, who just started his lab at WashU, Bob and Hopi, as well as Manea and Glory, and the Datta and Hoekstra labs. Thank you.
Time for a few questions.
Hi. Thanks for the talk, that was great. I was wondering if there was any anatomical difference between the mouse, and if so,
yeah, so the mice actually, great question. The mice actually differ in almost an order of 4. So the smallest mice and the largest mice are 4 times heavier. So what we do is we collapse them into the single morphology of a C57 lab mouse.
Thank you.
Beautiful talk. Do you think the differences would get bigger or smaller if instead of being in an open field, they had some task? And would it depend on the task?
Yeah, I think that, you know, the next stage for this this set of questions is to try to produce task structure and to try to make the environment that they're in more interesting, add more affordances, and kind of sample their full syllabic repertoire, because of course we're not sampling from all the different behaviors that they could possibly produce. So I think that we would have a much expanded set of behaviors that they would produce. I think we would see much expanded differences. You know, the Peromyscus that we're looking at here, they're, you know, well known for differences and things like their burrowing. And I think we would see a lot of maybe ecologically relevant differences as we change the task structure.
Hey Tim, great talk. So it kind of looks like, correct me if I'm wrong, you're mapping on the syllabi of the deer mice to the house mice, and you're kind of assuming that those ethograms are sort of one-to-one mapping. Have you tried to like do the UMAP or do, you know, sort of this projection independently and look for, you know, are there regions where maybe actually this syllabus is conserved only in one of these 2? You know, looking for, instead of what are the mappings that are similar and enriched, you know, are there any unique syllabi?
Yeah, that's a great question. So something I kind of glazed over in this is that there's 2 ways in which we're doing these analyses. One is with the algorithm that our lab has kind of pioneered, which is the MoSeq algorithm, which sort of assumes that there's a set of syllables that all of the animals you're projecting into it share. We also have a second algorithm to syllabify behavior, which is sort of a model-free algorithm in which we're not making those assumptions. And so the analyses that I showed you, in which I'm comparing different species and subspecies of mice, are purely with that model-free algorithm. And the reason is for the reason that you just stated, which is that we don't want to make an assumption about them having a shared set of behaviors.
Thank you.
Let's thank Tim. Next speaker is Ueli Rutishauser from Cedars-Sinai Medical Center in Caltech. And the title of his talk is A Shared Code for Perceiving and Imagining Objects in Human Ventral Temporal Cortex.
Great. Thank you, everybody. Good evening. Thank you for the organizers for the invitation to speak. Today I'm presenting our project on the neural substrate of visual imagination. Before I start, let me note that I'm presenting on behalf of the first author of this work, Varun Wadia. Unfortunately, Varun was unable to be with us due to Canadian visa issues, so I'm going to try to represent him here. So mental imagery is a generative process that allows us to visualize images in the absence of input. Imagery plays an important role in many aspects of human cognition. To name just two examples, imagination allows us to bring back previous experiences from memory and to produce visual art and to produce non-existing animals, as we just saw in the prior talk. A long-standing theory is that the nervous system enables imagination by using the same substrate it also uses for perception. So it reactivates that substrate, much like in a generative model. So we set out to test this theory. We did so by recording single neurons in human subjects in the ventral temporal cortex. The ventral temporal cortex is well known to support object recognition and has been extensively studied both at the single neuron level, as is shown here on the left, a neuron from a macaque that responds only to faces, as well as in humans using fMRI, where classical results show that specific parts of VTC become active when you show houses, faces, or chairs, for example, to subjects. Now, the very same experiments also show that the very same part of human VTC become active when you ask subjects to imagine the same category of objects. What remains unknown, however, whether this means that the same neurons are activated. It could be just that they're co-localized, but doesn't mean that they share the same substrate. So we set out to test this theory, and the data I'm going to show you today comes from extracellular recordings in human epilepsy patients. The recordings are performed using microwire electrodes that extend at the tip of the depth electrode, and these depth electrodes are implanted in the human subjects to localize their seizure as part of their treatment. So the data I'm going to show you today comes from 16 patients, and in total we recorded 714 neurons across these patients. So we started by showing them a long series of 500 images repeated 4 times each. These images were chosen from the categories faces, text, plants, animals, and objects. And then we used this data to map out the tuning of the neurons, so we could later ask whether the tuning remains the same when we ask subjects to imagine the same stimuli. So first, we started by parameterizing the arbitrary objects using a deep neural network. We used AlexNet, and in the data I'm going to show you, everything is based on the FC6 layer of AlexNet. We reduced FC6 activity using PCA to 50 dimensions. So the representation we are going to be using is a 50-dimensional vector for every object in this space. So we first asked whether neurons in human VTC represent arbitrary objects using an axis code, much like they do in macaques, where this is a well-established fact. So to do so, we attempted to find a preferred axis for each cell, and the preferred axis is defined as explaining the most variance possible if you project the activity of each of all the stimuli that we show to the subjects onto that axis. Now, a non-trivial and very interesting prediction of this model is that there should be orthogonal planes to this axis, and all the images that lie on this orthogonal plane should result in the same response of that neuron. The neuron should not change its response, and we use this as a defining characteristic of a cell that has axis coding. So consider this example cell. The stimulus onset is at 0. It responds vigorously after about 150 milliseconds in a graded manner and that changes with the category that the image was picked from. Now I know most of you work in rodents, so I wanted to point out that this is considered a fast response in the human brain, and I've marked there also where the average human hippocampal cell would respond to this stimulus. So consider this cell from the axis tuning point of view. So here in the middle, each dot is an image. The color of the dot is the firing rate that the neuron fired in response to that image. And you see there is a gradient from left to right, but not from top to bottom. So that means this neuron is axis tuned as shown on the right. Now, it turns out this is by far not an outlier. In fact, we found that 80% of cells in the human VTC were axis tuned. And this means they have a monotonic increase of response along the preferred axis, as you saw on the left for all the cells, and they have flat tuning across all other axes, including the principal axis, but all 49 other axes are like that. Now, to study imagination, we need to be able to take as input neural activity and get as an output an image. What I have shown you so far is the opposite. I'm putting in an image and neural activity comes out. So the question is, can we invert this mapping to study imagination? And it turns out we can. We tested this in 2 different ways. The first way is we try to decode which image was shown by predicting from the neural activity the feature in FC6 space and doing a nearest neighbor search in FC6 space to see if we recovered the image that we actually showed to the subject or another image. And we could do so well. Now, this model sometimes picks a different image, and the different image that it picks provides insight into the neural code of VTC. So consider these images that we showed to the subjects and compare them to the images that our algorithm predicted that the subject saw. So these are images that they were not available to the subject. We never showed them to them. They're from a different data set. So notice that sometimes the semantic category of the image is very different, but visually they're very, very similar. For example, if you compare the pot with the cup. So this indicates to us that this code is visual shape-based rather than semantic. So armed with this new machinery and framework, we now set out to test the key question whether the same neurons activate when subjects encode an image or view an image, here the backpack, compared to when we ask the subject with a verbal cue to imagine the same object. So we say, imagine the backpack, describe to us how it looked. So consider this cell. This is another axis-tuned cell with the same plot that I showed before. We asked the subject to imagine the subset of these 8 images. And when they viewed the images on the screen, this neuron responded to this subset of images, mostly to the ring, the yellow image. And when we then asked the subject to imagine these stimuli, similarly, the neuron responded most to the ring and some less to the other images, and I will come back to that. So overall, we found that about 30% of the cells reactivated during imagination, and they did so in a stimulus-selective manner that is the same than during perception. So we showed this in the following way. So let's return to this cell. Now, imagination is slow. You can only imagine so many things. It takes time. So they can't imagine 500 stimuli. So they were only imagining these stimuli. But notice how we chose the images. We chose the images such that they either vary mostly along the preferred axis or mostly along the orthogonal axis. So this way we could test for each possible pair of images whether this prediction is observed using the axis that we established before using the perception task. And that was indeed the case. We found there's a strong correlation only on the distance along the x-axis, but not along the orthogonal axis and all other axes. So this means that these cells observe the same code during imagination than they do during perception. So we next put this model to the test. Does this actually allow us to reconstruct what subjects are imagining? And it turns out it does. So each line here is a single session, so these are not that many cells, just a few cells. And similarly to the perception, the mistakes that the model makes, if you will, indicate that the imagination here is visual, not semantic. So for example, these 3 images that one subject was asked to imagine, the reconstruction algorithm deciphered that this is the most likely stimuli that the subject had in mind. And you see here that for us, the top one looks very different semantically, but visually, really, it's not. It's the same visual code. So we believe that even during imagination, the code here remains visual. We further tested the similarity between the 2 codes using generalization analysis. So when we trained a decoder doing viewing and tested it to imagination, it generalized well. Didn't generalize perfectly, and one reason for that is what's shown on the right, is that this population does what many call reality decoding, which is it can differentiate between perceived and imagined stimuli across many neurons. It's a broad code. So one could presumably do much better generalization with sort of cross-task learning. But the one that I show here is exclusively trained on one task versus the other, so it has no information. So in conclusion, our data shows that human VTC neurons use an access code to represent the visual world. This code is recapitulated during imagination, and it allows the reconstruction of what subjects imagine. And overall, this data presents single neuron evidence for a generative process in human VTC that is the basis for imagination. So what does this all mean? What's the underlying mechanism that is engaged here? If we return back to one of our favorite diagrams in the textbook, the Feldman and Van diagram, we can sort of see that the perception process, the feed-forward process, we understand it fairly well, and I've talked here about the higher visual area, the VTC, the visual code. And I have now shown you that we believe that in VTC, the very same neurons are activated from memory when we ask subjects to imagine, thus leading to imagination of the backpack here. The big open question now is what activates this process? Where does this information come from? And that's something we are currently working on to figure out. So with that, I would like to thank first and foremost the first author, Varun Wadia, of this study, and all my collaborators. In particular, this is a collaboration with the lab of Doris Tsao and my clinical collaborators at Sinai that has made these recordings possible. Thank you.
Time for a few questions.
Yeah, thank you for this really interesting talk. One thing which wasn't clear to me was when you ask the subjects to imagine something, do you have they seen a specific image of what they should imagine, or are they given words like imagine a scorpion, but they can imagine any scorpion?
Yeah, that's a good question. No, we asked them to imagine exactly what they saw. We saw them recall from memory this backpack that we showed you.
Yeah. Thank you.
You claimed, I'm sympathetic to this claim, but you claimed that the tuning is visual and not semantic. But unless I miss something, you're filtering everything through this vision model. So I'm wondering whether I missed something or whether it could be just explained by the fact that everything's being driven by AlexNet. You know what I mean?
Yes, I did not mention, but this is true. First of all, what I did not mention is that you can produce the very same result with many other different vision models. In particular, we can also produce these results, and in fact, they explain more variance if we fit completely unsupervised models that were trained in a completely unsupervised manner, so no labels. So like these dino-type models. So yes, AlexNet has some semantic information, but we don't think that's what the neurons pick up on.
Okay, thanks.
Are there any systematic intersubject differences, for example, as a function of age or the severity of the epilepsy?
Yeah, that's a very good question, and we are not powered to investigate that kind of question. They do have a questionnaire where they tell us how well they're able to imagine, and those people all rated fairly highly on that ability. We haven't had someone yet who said, no, I can't imagine anything. That would be very interesting to study, but hasn't happened yet.
My question is related. I was wondering how many subjects there were, how many human subjects there were in each of those categories, and if you asked them whether their imagination was visual or semantic when they were visualizing those objects.
Yes, so we asked them, do they really describe it? They're like, oh yes, there's this man and it has a red nose and so on. We haven't really related that to the visual features. We did the standardized questionnaire on how well they can imagine, and remarkably, they all could imagine really well. So once we get somebody who can't, we'll hopefully get an answer to that.
We have one quick question.
Do you record those patients 24/7, and can you decode their brains? And do you have any patients that are lucid dreamers by chance?
Well, those are all good questions. Yes, we can record them 24/7. We have not attempted to decode dreams, but good idea. Thank you.
Thank you, Ueli. Next talk is an invited special lecture from David Anderson from Caltech in the Howard Hughes Medical Institute. The title of his talk is The Neural Coding of Internal States Underlying Innate Social Behaviors.
So I'd first like to thank the organizers for the opportunity to speak at this meeting. It's only the second time in I think at least a decade that I've spoken here, and it's particularly exciting for me given my lab's move into computational and systems neuroscience recently. I should say, though, that I find this an intimidating crowd to speak in front of, not only because I'm not a computational neuroscientist by training, I was trained in molecular biology and genetics, but also because I was talking to Tatiana just before this session, and she told me that she's become very concerned about the magnitude of line attractor scores, and that made me insecure that my line attractor score wasn't big enough for her. But hopefully, you know, you do what you can with what you have, and hopefully I'll be able to convince you that line attractors, or at least approximate line attractors, are useful for thinking about processes other than cognition. So I'm subtitling this talk from causation to correlation and back, because I was raised in the molecular genetics catechism that correlation does not imply causation. And so as you'll see, our studies of this subject began with causation, but it's taken us into representational and observational studies for what I think are good reasons. And but I hope I'll show you that we're trying to come back to causation in the end. So what do I mean by an internal state when I talk about internal states underlying innate social behaviors? We think about internal states by analogy to Kent Berridge's work on motivation as a type of low-dimensional intervening variable that integrates or combines a variety of different stimuli and computes some low-dimensional variable, which then fans out to control motor responses and bodily responses, as well as changes in input-output processing. And in the case of affective states, we may have a conscious experience of these states that we call emotional feelings. Now, internal states of the homeostatic type have been studied extensively, such as hunger, thirst, and temperature. And we know a great deal about the causal control of these states from work from many labs, including Scott Sternsen's and Yuki Oka's and others. But these also include affective states that are much less well understood, such as defensive states that we call fear, or aggressive states that we call anger or sexual arousal. Now, trying to find neural representations of affective states is challenging. In the case of homeostatic states, operant conditioning and reward learning-based assays have been used as direct measures of internal state variables, and that's allowed supervised analysis of neural data that is recorded during the execution of these learned behaviors. But if you do this in the case of innate behaviors, and many people have, where you look at the behavior, you measure neural data, and then you do a supervised analysis to try to identify neural activity that correlates with behavioral variables, you run into a problem, which is how do you distinguish the representation of behavior from that of very highly correlated internal states? So if we expose a mouse to a predator and it freezes, and we see a spike of activity in their hypothalamus, is that spike of activity encoding the identity of the predator, or is it coding a state of fear evoked by the predator? And that's the essential problem that I want to talk to you about today. and I'm going to describe an alternative approach that we've taken in my lab with the help of Scott Linderman at Stanford, which is to try to identify neural representations of internal states through unsupervised analysis of neural population dynamics. And then to take that back, the signals that we identify, and to see how it relates, if at all, to our measures of behavior carried out in parallel. Now, which populations do we collect these data from? To explain that to you, I have to take you back into the world of causation and the sort of what I consider the perturbation experiment in the field of affective states, and that is Walter Hess's demonstration in the late 1920s that the injection of current into the medial hypothalamus of a cat could evoke a state that he called defensive rage. And that was almost indistinguishable from the expressions that were evoked if the cat was exposed to a natural threat of some type. And the reason that this was important is that Hess did not observe these reactions when he performed this experiment in other brain regions. And so that falsified the hypothesis at the time that affective states or emotion states were sort of diffusely distributed around the brain and indicated that there were specific loci for those states. Now, it's taken 70 to 80 years to go from those original demonstrations to actually identifying the cells that are responsible for this activity. First, for those of you who may be unfamiliar with the hypothalamus, I just want to put the part of the hypothalamus that has stimulated in context. It lies in a region that's called the medial hypothalamic behavioral control column. This is a group of highly interconnected subcortical nuclei characterized by Larry Swanson and his colleagues, mostly in the rat. And these columns between them, these nuclei, receive input from upstream regions like the bed nucleus of the stria terminalis and the medial amygdala, which in turn are activated by olfactory and other types of cues. And so these nuclei together control the behaviors that we refer to in my lab as the 4 Fs: feeding, freezing, fighting, and mating. And within this control column, I'm glad somebody got that. I'm going to be focusing most intensely on VMH, the ventromedial hypothalamic nucleus. And that is the region that worked from Menno Krook and his colleagues in the Netherlands in the 1980s in rats using very precise focal stimulation experiments identified as what they called the hypothalamic attack area, a region from which you could evoke attack from a rat towards a conspecific. Now, we wanted to move this field into mice so we could take advantage of the genetic tools for marking and mapping and manipulating various neural circuits. And we were stymied at first, and we is Dayu Lin, who was a postdoc in my lab at the time. And we were stymied because the experiment doesn't work in mice. If you stimulate VMH electrically in mice, you get freezing and jumping, but you don't get any attack. And so we were ready to throw in the towel on studying brain stimulation-evoked aggression in mice when I learned about the advances that Carl Diceroth and Ed Boyden had made in using optogenetics to stimulate deep subcortical regions, and I suggested to Dayu that she try this experiment. And although she was initially quite resistant to trying it, she dutifully carried out the experiment. And this was the result that she saw. Many of you may have seen this video where we are now optogenetically activating neurons that are anatomically localized in the ventrolateral subdivision of VMH, VMH-VL. And you see that we can evoke essentially time-locked attack to an inanimate object as well as to other mice, male mice, female mice, etc. Now, it took us about 3 years from that time to identify genetically the actual neurons that were carrying out or responsible for this brain stimulation evoked aggression, and we did that using CFOS and correlating that with a number of molecular markers, and identified the type 1 estrogen receptor as a stable marker for neurons that are preferentially activated during aggression. And independent work from Nirao Shah had identified the progesterone receptor as a marker for cells activated during these social behaviors. And so we generated estrogen receptor Cre mice. So these are mice in which Cre recombinase is expressed under the control of estrogen receptor regulatory units. And then we inject into VMHVL a Cre-dependent virus that encodes channel rhodopsin. And we found that when we stimulated only these estrogen receptor positive neurons within VMHVL, we could trigger time-locked aggression to both castrated male and female intruders. And importantly, when we inverted the experiment and stimulated the non-estrogen receptor positive neurons, we did not observe that effect. So this is not a random correlation, but specific to those cells. Now, interestingly, as we played with the parameters of optogenetic stimulation, we observed an unexpected phenomenon, which was that according to the intensity of stimulation or the density of labeling of these neurons with channel rhodopsin, we could evoke different types of social behaviors. So weak stimulation evoked mounting, moderate stimulation evoked a mixture of mounting and attack, and strong stimulation evoked attack. We originally thought that this mounting was sexual in nature, but more recently we've done experiments that showed that it is a low-intensity form of aggression, because during sexual mounting, male mice emit ultrasonic vocalizations, and during this agonistic mounting, they don't. So over the years since that time, work from my lab and from many other laboratories, especially work from Catherine Dulac on neurons in the medial preoptic area that control parental behavior, and more recent work on mating neurons from my lab and others, have led to a picture of the hypothalamus as a collection of highly specific cell types that are apparently dedicated to the control of specific behaviors, and that's consistent with the view that the behaviors that are correlating, that are controlled by the hypothalamus, are encoded by some sort of labeled line system. So not much interesting to you out there in the computational and systems neuroscience community. But as we looked at the accumulation of these data, it became clear that this was not a particularly satisfying explanation, although I still argue with my hypothalamic colleagues about this. But for example, stimulation not only of VMH, but also a number of its upstream and interconnected areas can all evoke attack optogenetically. This is work from my lab and many other laboratories. So if stimulating these nodes can all produce the same behavior, why do you have all these different nodes? And that raises the question of what information is being represented in each node. As I said earlier, is it sensory cues, object identity, internal state, premotor programs, action selection? We don't know, and it's critical to understand that so that we can then understand how these representations are transformed as information flows through the circuit, and not just in a feed-forward way, but there's extensive feedback in the circuitry as well. And so as a result of that, we decided it was important to begin to study the representations, the activity of these so-called hypothalamic attack neurons that we had discovered in freely behaving mice under naturalistic conditions. And we had taken an initial stab at that with Dayu and managed to do some single unit recordings with tungsten wire bundles, but that was not easily adaptable to recording from genetically marked cell types. So we were early adopters as a result of the miniscope technology developed by Mark Schnitzer at Stanford, which allowed us to express GCAMP, genetically encoded calcium indicators in the estrogen receptor positive neurons, the same population that can produce attack when stimulated. And we found, fortunately, that mice with these miniature microscopes that weigh about 2 grams mounted on their heads are able to adapt to them and to carry out attack and mating and investigative behaviors. So the results that we got from those initial experiments gave us a surprise. And that is, whereas the perturbation experiments might have led one to expect that you would see time-locked activation of neurons that is coincident with the onset and offset of attack bouts, and that is highly specific to attack behavior as opposed to sniffing or mounting, we saw very few neurons that fit that profile, recording from hundreds of these cells. Rather, what we found is that most of the neurons were not time-locked to attack onset and offset, and when we used a measure called choice probability to estimate the tuning or the preference of neurons for different types of social behaviors, we found that only a small fraction, between 3 and 10% of these neurons were selectively tuned to attack versus sniffing, with the majority of them exhibiting mixed cell activity. Nevertheless, we were able to train linear decoders to correctly predict with a fairly high degree of accuracy attack and sniff behavior. So there's some sort of information in the population activity that is relevant in some way to the behavior. And so that, in turn, raised the question of whether we could extract behaviorally relevant signals by analyzing the dynamics of estrogen receptor neuron population activity. And to do this, we had a multi-pronged collaboration that included using beautiful experimental data generated by Tomomi Kirigo at my lab, now at Johns Hopkins, and initially by Ann Kennedy, a computational postdoc in my lab, now at Scripps, and then increasingly by Aditya Nair, and Adi is here at the meeting today, and as you'll see, he's pushed this project forward. All of this was critically enabled by work that Scott Linderman at Stanford has done to develop unsupervised probabilistic methods for modeling dynamical systems in neural activity, in neural data. And basically, the method that he introduced, called recurrent switching linear dynamical systems, approximates a nonlinear system as a sort of concatenation of linear dynamical systems, and that's done by first reducing the dimensionality of the data into a latent factor space, and then asking the algorithm to find a certain pre-identified number of substates within the state space, and then the algorithm fits a dynamical system model to each of these substates and learns the appropriate parameters and then stitches together a linear dynamical system model that can then either be visualized as a 2D flow field plot of the first 2 principal components, and that can be converted to an energy landscape. I'm sure I'm not telling any of you anything you don't already know. So what do we see when we apply this approach to this data set? And so we get a model of the form dx dt is ax plus bu, where A is the learned dynamics matrix, x are the latent factors, u are the inputs. And the interesting thing about this, as I learned from Scott and Adi, is that dissecting the properties of the dynamics matrix can reveal some interesting computational properties of the system, because the eigenvalues of the dynamics matrix can be converted to the time constants of the different dimensions in the latent space. And you have no idea how long it took me to rehearse that phrase in front of the mirror so that I could say it properly. Thank you. So again, not telling you anything you didn't already know, but if all of the dimensions of this latent space have short time constants, it suggests that activity along these different dimensions relaxes very quickly following an input along that axis. But if you find that one of these dimensions has a time constant that is significantly longer than the others, then it suggests that the system has a integrator property or a line attractor property. And so it wasn't a foregone conclusion that we were going to find this when we applied the RSLDS modeling to our data set, but that is in fact what we found. And here are the different dimensions, the time constants of the fit dynamical system from half a dozen different mice. And you can see that the first dimension is much longer in its time constant than the others, upwards of 100 seconds, sometimes more. And if you plot this in neural state space as a flow field, what you can see is a line of slow points, this white region over here, which looks like a line attractor, and you can see the trajectory of the neural population vector entering into this region. And if you now superimpose on that the scoring of the actual behavior, which was recorded simultaneously on video, what you find is that the system enters the attractor as the animals start to interact and sniff each other, progresses along the attractor through dominance mounting, and goes to the very end during attack. And in a 3D energy landscape visualization of the model, you see it looks like a sort of trough or gully in neural state space. And particularly interesting aspect of this to us is that if we remove the first intruder mouse and then put in a second intruder mouse, the activity is transiently kicked out of the system, but returns very quickly as soon as we reintroduce that second mouse back to the place that it was when we first removed the initial mouse. And so that looks sort of like the type of relaxation you would expect from an attractor-like system. Now, if we plot the weighted average of activity of the neurons that are weighted on this slow mode or integration dimension and align that with our behavioral annotation, you see that activity ramps up slowly as the behavior progresses from bouts of sniffing to dominance mounting in blue, and then flattens out and stabilizes when the animals are attacking. And you can decode very accurately, you can train two-way decoders to distinguish the type of behavior the animal is showing from the magnitude of activity in this dimension. But more importantly, you can also decode interbout intervals, these white regions that are between different types of behavior. So although to your human observer eye, you couldn't tell looking at this interbout interval whether the mice were in between an attack bout or were in between a sniffing bout. You can actually decode what behavior these interbout intervals were between from the magnitude of this integration dimension. And that's because, as you can see, the activity doesn't really dip down in between the bouts of attack. And so that suggested to us that perhaps this approximate line attractor might be encoding an internal state of aggressiveness, such that as activity moved along this attractor, the intensity of the aggressive interaction escalates. And that would account for the observed scalability or variable intensity of the state, and the ability of the system to be moved out and to relax back in would be a reflection of the persistence or the stability of this internal state. And this was interesting from a sort of theoretical perspective that Ralph Adolphs, my colleague at Caltech, and I have written about, where we suggested that the evolution of emotions may have proceeded through what we call emotion primitives. These are properties of behaviors and their underlying internal states that distinguish these state-dependent behaviors from simple stimulus-response reflexes. And two of the important ones are scalability and persistence. And so it was very interesting that this unsupervised approach revealed a potential mechanism that could encode both scalability and persistence of an internal state. Now, the other thing that observation that made us think there was something real and biological here was the following. So the advantage of Scott's approach is that you can make these models on single trial data from freely behaving animals. The disadvantage is that there is a lot of individual variation in the innate behavior of mice in aggression. And so we tried to ask whether there was any relationship and turn this bug into a feature, and we plotted the time constant of the slow mode dimension against the level of aggressiveness of these mice measured independently, and you see that there's an extremely strong correlation between the two, across over a dozen mice. And what that suggests is that the level of aggressiveness is proportional to the stability of the attractor, such that attractors with longer time constants are found in mice that tend to attack more, whereas attractors with shorter time constants in the slow mode are found in mice that tend to attack less. Now, just to try to convince you that this isn't some artifact of GCamp imaging and is what you would see if you recorded in any region of the hypothalamus, we performed the same type of modeling on recordings that we had made in neurons in a neighboring region of the brain, medial preoptic area, which we had shown causally control mating behavior. And there, we found that there was no evidence of a slow mode or line attractor dynamics. Instead, the population vector displayed rotational dynamics around a trivial point attractor located at the region where the animal is inactive. And this, the angle of the rotation corresponds to the progression of the behavior, the mating behavior, through discrete stages where different behavior-specific populations are active, like sniffing, mounting, and intromission. And we don't see that type of behavioral specificity, as I said, within VMHVL during attack. So even though mating and aggression are two very similar social behaviors, sort of two sides of the same coin, and even though the optogenetic phenotypes of activating or inhibiting the respective populations are identical at the level of neural representation, these two brain regions seem to use completely different strategies or mechanisms. And we don't know why yet or how general this is, but one possibility, simple model, which I call the television or radio model, is that VMH controls a continuous variable that is related to the intensity of an internal state, whereas MPOA may encode the selection of a particular action, and within each action you can variably control the intensity of the state. And that, interestingly, correlates with different cytoarchitectonic and biochemical features of the neurons that make up MPOA and VMH. The neurons in MPOA are 80% GABAergic, whereas in VMHVL they're 80% glutamatergic and have this core shell construction. And whereas Catherine Dulac's work has shown that behavior-specific neurons make non-collateralizing projections to specific downstream targets, our own experiments have shown that there's a great deal of collateralization of individual neurons to a multiplicity of downstream targets, consistent with more of a broadcasting type of function. Okay, so that's a model. It shows us that there is a representation of what we think is an internal aggressive state, but I was rapidly disabused of my enthusiasm for this initial result by colleagues in computational systems neuroscience who kept asking me, well, how do you know it's not just inherited from an upstream structure and passively projected onto VMHVL, or how do you know it doesn't just represent ramping sensory input? And so to answer that question, it was necessary to try to design and execute perturbation experiments. And up to that time, The only type of perturbation experiments of continuous attractors that had been performed was in the ring attractor that encodes head direction in Drosophila, which has been beautifully studied by Vivek Jayaraman and Gabi Maiman and others. And Vivek and Johannes Selig famously did experiments where they could activate a region next to the existing bump of activity and show that even after the offset of activation, the activity would persist in that new bump and then move over to another position around the ring. And so we thought, wouldn't it be nice if we could do analogous experiments for this approximate line attractor? And by the way, you'll forgive me if I don't always use the word approximate as a prefix to line attractor. It's been beaten into me that unless the eigenvalue of the attractor dimension is 1, it's not a real line attractor. Our eigenvalues are 0.99 something, but I'm still calling it an approximate. So from now on, you'll forgive me if I leave out the approximate. And so the problem is that in order to do those types of perturbation experiments, you need a head fix preparation. And head fix mice, as you might expect, don't fight. They don't even growl, as far as we know. And so it seemed like a non-starter of an experiment until Adi, my computational graduate student, paired up with a very talented experimental postdoc, Amit Vinograd, who's also here at the meeting and is going to be talking in the workshop at Mont Tremblant this weekend. And they came up with a solution. And the solution, I should say, I have to credit a very important breakthrough made by my former student, Nirav Shah, at Stanford, that had nothing to do with wanting to investigate line attractor dynamics. What he did was compare activity in VMHVL using FOSS mapping initially of mice that were engaged in aggression and mice that were watching two other mice fight. And he found that there was a high degree of overlap in the neurons that were activated during engagement of aggression and observation of aggression. And he named these populations mirror neurons for aggression. He was able to show with some activity-based labeling techniques that the neurons that were activated during observation of aggression could actually suffice to promote attack if they were reactivated optogenetically. And so we wondered if these mice that were observing aggression might also exhibit line attractor dynamics, and if so, might they exhibit it not just as freely behaving animals, but also in a head fix preparation. And so we fit RSLDS models to activity recorded from mice engaging in and observing aggression. And to our delight, we observed line attractor dynamics in both systems. And there was a high degree of overlap in the neurons that were weighted on the attractor dimension by the RSLDS model. And so the next step was to see if these line attractors could be instantiated in a head-fixed mouse watching two intruders fight, and they could. In this case, Adi and Amit used a two-dimensional model, and what they did was a sort of manual closed-loop experiment where Adi sat in the two-photon room with Amit. They collected the data. He fit the RSLDS model and then identified the neurons that were weighted on the attractor dimension, which is shown in green, the X1 dimension, the non-attractor dimension shown in red, which is X2, the orthogonal dimension, found those neurons in the imaging field, and then we used holographic two-photon optogenetics to selectively activate either the X1 or the X2 neurons. And as predicted, activation of the X1 cells that are weighted by the model, and I should say, by the way, this activation is done after removing the demonstrator mice from the room. So we're asking whether by directly activating these cells, we can observe integration and movement along the attractor in the absence of any natural input. And as you see, we could. activating the X1 neurons allowed us to move the population vector to arbitrary position along the slow points here, and that if we followed that by an off-manifold perturbation stimulating the X2 neurons, the activity relaxed back to the region where the vector was before we did this activation. And these data were recently published, so you can read all the gory details, but I also invite you to see Amit's talk at the Causal Perturbation Workshop at Mont Tremblant if you're going. Now, having seen this really was a game-changer experiment, at least for me, because it suggested that we were looking at a real biological entity that was instantiated in the region that we were observing and not just inherited from an upstream region, although the data don't exclude some mesoscale feedback with interconnected regions. But Amit next wanted to use the power of the system to ask something about implementation, specifically line attractor models like that proposed by Sebastian Chung initially to explain eye position, assume that the persistent activity that gives rise to the slow mode reflects recurrent connectivity between the neurons that are contributing to the circuit. And so Amit tested that now by performing single neuron stimulations, stimulating either individual X1 or X2 neurons, and then simultaneously imaging activity in other X1 and other X2 neurons in the preparation. And what you can see here is that optogenetically activating a single X1 neuron produces integrative responses in other X2 neurons within the imaging field, but does not produce those responses in X2 neurons, which are weighted on the orthogonal dimension. Conversely, activating the X2 neurons does not recruit other X2 neurons, but it does exert some influence on X1 neurons, which is an important result for thinking about attractor dynamics. So this suggested that the implementation of this approximate line attractor involved, at least in part, some sort of local connectivity or recurrent connectivity selectively among the attractor-weighted neurons in VMHVL. And again, we took advantage of the individual variability in this system to ask a correlative question. You recalled I told you earlier that freely behaving mice that are very aggressive have longer time constants than less aggressive mice. Now, we can't measure the aggression in the head-fixed animal, but we can measure the decay constant of the attractor once we've identified it. And then, so we did that for a number of individual mice, and then we also, in those mice, measured the strength of the connectivity between each of the X1 or X2 neurons and its follower cells by stimulating and then imaging from the other cells and then measuring that either as the peak activity or the area under the curve. And when we plotted those two variables against each other, there was a remarkably strong correlation between the decay constant and the strength of the connectivity, such that animals that had a faster decay constant had a lower degree of functional connectivity, whereas animals that had a slower decay constant had a higher degree of functional connectivity. And again, this was not an expected result, but again, it suggests to us that there's something real and biological going on here. So what I've told you here is that these estrogen receptor neurons in VMH display approximate line attractor dynamics during aggressive interactions, that the stability of the attractor scales with aggressiveness across individuals, that these dynamics are instantiated by ESR1 neurons in VMH, not inherited, and that the attractor-weighted neurons exhibit selective functional connectivity. And functional connectivity strength scales with attractorability. So this made us interested in understanding more about implementation, specifically the source of the persistent activity that gives rise to the slow mode. And as many of you know, persistent activity can arise electronically through recurrent connectivity, but it can also arise biochemically through peptidergic neuromodulators or biogenic amine neuromodulators that act through G-protein coupled receptors. And interestingly, when Adi made a mechanistic RNN of this system, in order to produce a network time constant that was consistent with our observed data, he had to invoke synaptic time constants on the order of 20 seconds, which is more in the realm of a G-protein coupled receptor-mediated signal transduction. If he tried to do this with fast glutamatergic synaptic time constants, it didn't show these properties. So this raised the possibility that maybe neuromodulators were playing a role in this slow dynamics, and our single-cell RNA sequencing has shown that these neurons express many different G-protein coupled receptors, including receptors for oxytocin and vasopressin, which are two neuropeptides that are well known to be, play a role in social behaviors. And so to ask whether these receptors might play a role in aggressive behavior, which wasn't previously known, and also in attractor dynamics, George Muntoufaris, a molecular biologist in my laboratory, also dragged kicking and screaming like me into systems neuroscience, developed a method to simultaneously express GCAMP and to use gene editing to functionally disable receptors for both oxytocin and vasopressin in the same neurons in adult animals. And that allowed him to get both a behavioral phenotype from these animals and a physiological phenotype from these animals. And while there was very little overall effect on the overall level of activity of these neurons during aggressive behavior, when we fit the data with RSLDS models, we saw that the line attractor had virtually disappeared and was converted to a point attractor. And you can see here that the integration of activity that occurs across the attack bouts is not seen when these two receptors are knocked out or knocked down. Rather, you have spikes of activity that return quickly to baseline. So the integrative properties of the slow mode seem to be dependent on some way on these two receptors. Whether the receptors are required just to initiate these dynamics or to maintain them, we don't know. But they are consistent with the RNN modeling that Adi did and suggest that slow neuropeptide signaling may be involved. And then finally, and those data also have been published recently if you're interested in learning more. Finally, we wanted to ask whether this idea generalized to a different type of social behavior. And so Adi teamed up with a graduate student, Mengyu Liu, who had discovered female-specific neurons in VMHVL that control sexual receptivity and lordosis behavior. And she imaged these neurons throughout the estrus cycle and discovered that during mating bouts, there was ramping activity exhibited by these neurons, which produced an RSLDS model that showed a region of slow points along which the population vector moved up as mating proceeded from mounting to intromission to ejaculation. And Mengyu was able to carry out, amazingly, what I think is the first perturbation of line attractor in freely moving females. This is not holographic stimulation, it's wide field general inhibition, but you can see that this pushes the population vector out of the attractor and it relaxes back to the original population. So these data, and then finally she was able to show remarkably that if she continuously imaged across the estrus cycle, this line attractor is only present in the phases of the estrus cycle where the females are sexually receptive, and on non-receptive days it goes away and then it comes back again. And she was able to show with ovariectomy and hormone priming that the line attractor is hormone dependent, as you might expect. And finally, by collecting data, again, across many different individuals and many different trials, she tried to correlate quantitatively the degree of receptivity with the level of activity in this slow mode, measured as the area under the curve or how far up the attractor activity moves. And as you can see, there's a reasonably good and highly significant correlation between how far along the attractor the population vector has moved and the percentage of time the animals spend performing lordosis. And so that suggests that this line attractor may encode the level of sexual arousal or sexual receptivity in the case of females. So taken together, what these data suggest is the concept that in the hypothalamus that approximate line attractors with slow dynamics may encode internal affective states that underlie innate social behaviors, And interestingly, in both cases, we have evidence for modulation of these dynamics by hormones and neuromodulators, estrogen in the case of the female attractor, neuropeptides in the case of the male attractor. So these are our principal findings. I've shown you data on representation, instantiation, implementation, mechanism, and generality. And while this is a huge amount of work done by the people in my lab, we think it has important significance and, of course, raises a number of important questions for the future. First of all, it suggests that line attractors may represent internal affective states that underlie innate behaviors, at least, in addition to cognitive variables, expanding the potential functional role of this computational mechanism. We provide causal evidence that a line attractor reflects the intrinsic dynamics of the observed neural network, and I think this is the first causal evidence of a continuous attractor in the mammalian brain. It shows that line attractor dynamics can emerge in the hypothalamus and are not a special property of more recently evolved regions like corticohippocampal circuits. It's the first evidence of neuropeptidergic transmission underlying any type of attractor as far as we know. And as I said, we think it may be a general mechanism for encoding persistent and scalable internal affective states. And so among the new questions that this has saddled us with is, how are these attractor dynamics implemented? What's the relative contribution of this apparent recurrent connectivity and the role of the neuropeptides? Can we show that movement along the line attractor is actually causal for the behavioral state? That's still a correlation, and that's something that we're really desperate to know. How is the attractor read out and transformed into behavior? This matters for us as observers. It's not clear yet that it actually matters to the brain. And finally, does the attractor coding of internal states generalize to non-social innate behaviors? And I'll just share with you briefly a figure from a paper that was just published by another graduate student in my lab, Kathy Chung, with Adi, and she studied a complementary population of neurons in VMH that are in the dorsal region. They don't express estrogen receptor, and they react to predators. And what she found by fitting RSLDS models is that there is also a slow mode in the pattern of population activity in response to a predator. And remarkably, the time constant of that slow mode across individuals varies in proportion to the level of fear measured behaviorally as by how much time the animals freeze or run away in the presence of a predator, such that mice that have a lower level of fear show a faster decay down this attractor, and those with a higher level of fear or anxiety show a slower rate of decay. And this potential implication of line attractors in representing an anxiety state is of interest because it's been suggested by a number of people, Ed Rolls, Helen Mayberg, Kay Tai, and others, that depression, which is well known to be influenced by anxiety and stress, may represent a state in the brain that is a type of attractor, not necessarily a continuous attractor, maybe a point attractor. But the point is that this has been speculation, and this system may give us traction, experimental traction on that problem. And then finally, I'd just like to emphasize what I think is, what I've learned is the value of this manifold level of understanding, as Tatiana describes it, in between the sort of circuit neuroscience that's been going on now for over a decade, which emphasizes cell types and their causal roles and their connections, and the mesoscale properties of complex systems like the hypothalamus, and that this intermediate level may provide a way to bridge these two different scales. And also, I'd like to suggest that this hypothalamic system may provide a convenient and attractable experimental platform for understanding how these emergent properties, such as continuous line attractor dynamics, emerge from the cellular and molecular and connectional properties of the underlying neurons. And that's something, again, that Tatiana has written about and stressed the importance of. And so with that, I'd like to conclude and thank the members of my lab that I've already mentioned for their tremendous contributions to this work, especially to single out Amit and Adi and Scott Linderman, especially without whose steadfast collaboration none of this would have happened. And so with that, I will thank you for your attention and be happy to take any questions.
Hey, that's amazing work. I have a question about the line attractor. Sorry, I have a question about moving towards aggression or away from aggression vis-a-vis thinking about the line attractor. Is one way easier? What do you observe for like decay dynamics when nothing's happening, et cetera, et cetera? Does that make sense?
Yes. So if I can rephrase your question, I think what you're asking is the time constant of the ramp up that we see at the beginning of aggression similar to the time constant of the decay when aggression is concluding?
Yes, and additionally, if you were to conjecture, if you were to have the causal experiments, would you expect that moving one way or the other was equal? Or, yeah, basically.
We haven't done the causal experiments, but we have some experiments from another project where we've looked at the influence of starvation on aggression. It makes hangry mice, and there what we see is both an increase in the rate of ramping up and also a slower decay. And so while we haven't done all of the math to see if the same decay constant can contribute to both, the increased rate of ramping could be due to an increased rate or magnitude of input with the decay constant the same, or there could be a different decay constant controlling the ramp up and the decay, and that's something that we're going to be investigating in the near future. Good question, thanks.
Hey, yeah, over on the other side. Thank you so much. It was a really wonderful talk. I'm curious about the loadings of the neurons on the slow mode and the orthogonal mode.
Yep.
Is it really strongly mixed selective where the same neurons are involved in both? And you know, what drove this? I know you guys had to do the holographic, and I'm wondering if that was to target neurons or to...
No, we've looked at that question, and the representation of behavior selective and non-selective neurons weighted on the attractor dimension is about 50-50, if I remember correctly. The neurons were selected based on their physiological properties. Both the X1 and X2 neurons express estrogen receptor. Otherwise, they wouldn't be GCAMP positive. We know from our single-cell RNA sequencing that there are about half a dozen different subtypes of estrogen receptor positive neurons. So we're in the process of trying to understand whether those different neurons weighted on the different dimensions are molecularly distinct from each other or not.
All right, sorry, just to clarify, is it different neurons on X1? Is there a set of neurons on X1 and a set of neurons on X2? Or is it patterns of neurons where the same neurons involved?
There is some overlap, but there is largely distinct sets of neurons in X1 and X2.
Hello. My question was also about calming, the time constant for calming down, and whether it's related to the second eigenvalue. You know, because in people, I think there are two uncorrelated variables: how quickly a person gets angry and how quick.
Yeah, it's an interesting question. If anything, I would say our data are pointing to it being the opposite, that the second eigenvalue is related to the initiation of the process, because we think that that may encode some sort of a neophobia or arousal or novelty response. And depending on the setting, that will drive the population vector at an angle to the actual region of slow points. And then after the interaction is over, the decay occurs right down the line attractor dimension, where there's very little activity from the X2.
Okay, thank you.
Yeah, so this is the behaviors that you study are these innate behaviors that, although innate, they are time courses over a few seconds, right? So they develop, they have some temporal evolution, which would be enough time for the hypothalamus to interact with lots of other regions. So how do you take this, amazing, beautiful work that you've done and to a spatial temporal context that actually can take into account interactions with other areas?
Yep, that's an excellent question. There's a couple of approaches that we've taken. There's a data set on bioRxiv that hasn't been published yet, where we've recorded across 20 different nuclei in the hypothalamus using a multi-pronged silicon probe and looked at the correlations between activity and behavior. But with respect to this particular question and system that I'm talking about, where we're focused and we're led by data is the peptides oxytocin and vasopressin are not made in the region of the hypothalamus that show the line attractor. They're made in other hypothalamic nuclei that send projections to VMH and that receive projections from VMH. So one of the things we're interested in seeing is whether the apparent local functional connectivity revealed by the single-cell holographic experiments actually reflects some sort of mesoscale feedback loop, similar to what Karl Svoboda and his colleagues have seen between the cortex and the thalamus.
Thank you.
Yep. We're running a little late, so maybe one more question, and then maybe you can get David to answer the rest of the questions offline.
Sure.
Hey, thanks for the really nice talk. I was wondering, so it might be a very noob question, but these neurons are estrogen neurons, right? But you haven't actually talked about the role of estrogen in the behavior that you're seeing. But at the end, you kind of have an effect of the cycle on the stability of this line attractor. And I wonder whether you think this is related to the level of estrogen. How is this related? And if you could say a little word about this.
So just to separate, the effective estrogen that I showed was in the case of the female line attractor, and there we know there are naturally fluctuating levels of estrogen over the reproductive cycle. The male aggression neurons are also estrogen receptor positive, which may seem counterintuitive given the sort of pop neuroscience view that estrogen is for good things and aggression is a bad thing. But others have shown, not us, that estrogen receptor is necessary for full aggressive behavior in adults. People have knocked out the estrogen receptor in adult mice and shown decreases in aggressive behavior. It's also known that developmentally, the postnatal surge in estrogen that recurs around 2 days after birth is absolutely critical for masculinization of the circuits that control aggression. That's work of Niraj Shah and others.
And how is this in female?
In female, the female architecture seems to be what you get in the absence of the neonatal masculinizing effects of estrogen. There's, of course, an important role for estrogen in the functioning of these neurons in the adult, and there's work dating back to the late 70s and 80s from Donald Pfaff showing that estrogen can affect the excitability of female estrogen receptor neurons in VMH. And the exact relationship between what the hormone is doing to the cell and its activity and the circuit and how it's doing that is very interesting, and it's something we're trying to get at.
I see, thanks.
See you tomorrow morning.